Okay, let's revisit the various parts of the vascular system, and in particular we're going to start off by looking at the aorta, which is something that you've looked at before. And in the image that we see here, this is an aorta that, as you've seen before, has been stained with a special stain to demonstrate elastic fibers and elastic lamellae. This time around you need to pay attention to the fact that there are three layers in the wall of the aorta. Tunica intima, which is the layer closest to the luminal surface. The arrowhead is going around the luminal surface just now, and we can't really make out a clear distinction between the tunica intima and the middle layer, the tunica media, which is this layer here at this magnification, although I can see just a slight discontinuity in staining along here that indicates where that boundary is. So tunica intima, tunica media, which is made up of smooth muscle, which is sandwiched between layers of collagen and elastic fibers, and a tunica adventitia, which is the external connective tissue coat. And we'll increase magnification now, and we'll take a look at each of these layers in succession. So at higher magnification, this is the luminal surface here along which blood flow will occur, and this is lined by simple squamous endothelium. The cells, the nuclei of the cells at least, some of them bulge into the lumen. We can see these here. They're quite small. Now it's very difficult in an elastic stained specimen to see the exact boundary between the tunica intima and tunica media in a large elastic artery like the aorta, because the internal elastic lamina is not quite as distinct as it would be in smaller muscular arteries. But broadly we could say that the tunica intima extends from the pointer to about here. And it's here where we start to see the first of these elastic lamellae increasing in number as we progress through into the tunica media proper, which is extending from here to, say, here. And here we see the very large elastic lamellae. We see very little detail between the elastic lamellae other than some reasonably homogeneous pink staining, which really represents collagen fibers and smooth muscle cells. And we can in fact see stained the nuclei of smooth muscle cells here. And in an aorta we would expect to find perhaps 40 or so layers of smooth muscle with elastic lamellae sandwiched between each of these layers and with all embedded in collagenous connective tissue. If we move out a bit further, we find the outer boundary of the tunica media where the muscle ends, and then we move to the tunica adventitia, which is the external connective tissue layer, and which you'll have looked at before. Here we see individual elastic fibers embedded in collagenous connective tissue. The collagenous connective tissue on the aorta like this is quite dense. It's quite a thick connective tissue coat, and in fact in some senses almost resembles tendon, but tendon by definition is dense regular connective tissue, and only tendons are dense regular connective tissue. So we would refer to this as a dense irregular connective tissue external coat. Within this dense irregular connective tissue we'll see other small blood vessels. These are called the vasa vasorum, the vessels which supply the vessel, and you'll also see these gaps or openings here which are adipocytes or fat cells which are present in the connective tissue. We'll also see little patches of peripheral nerve, as we can see here, and peripheral nerve, the peripheral nerves that are found in the walls of the larger blood vessels are called nervi vasorum, which means nerves of the vessel. Again, we'll see here's an arteriole. I can tell it's an arteriole because it has an endothelium and only one layer of smooth muscle surrounding it. Anything less than 5 layers of smooth muscle is an arteriole rather than an artery. And you can spend some time looking around the wall of the vessel and observing what you can see. For example, there's a venule right here. And also you should go back to your original connective tissue set of notes and examine the slide that you looked at of aorta, which is stained with H&E rather than with stain for elastic fiber, and compare that with an aorta stained for elastic fibers. Here's an interesting slide, because on this slide we have a medium-sized muscular artery, the vein which would accompany a medium-sized muscular artery, and in fact we have both of these sections shown here and here stained with hematoxylin and eosin, as you can see on the upper one, and then an equivalent identical section stained to demonstrate elastic fibers on the lower section. So let's begin first by looking at the hematoxylin and eosin stained muscular artery. The tunica intima will be on the luminal surface somewhere here. The bulk of what we see here is the tunica media, which will be made up of circumferentially oriented smooth muscle, and the material which we see out here is connective tissue, which forms the tunica adventitia. Clearly it should be evident to you that the largest of the layers of the wall of an artery is the tunica media. Over here we see an equivalent vein that would accompany this. We can see that the vein, having a much thinner wall, has collapsed, and if this were expanded out, we'd see that the vein would actually have a larger lumen than the artery does and a considerably thinner wall. At this magnification it's difficult to make out what the wall structure of the vein is. It has a tunica intima on the luminal surface, it has a thin disorganized tunica media, and the bulk of the wall of the vein is made up of tunica adventitia. So let's first look at higher magnification at the structure of the muscular artery. Here's the muscular artery at a slightly higher magnification. The endothelial cells that line the luminal surface lie along here, and we can see occasional nuclei, very dark dot staining things, which belong to these endothelial cells. The tunica intima is thin, and here is represented by the mauve staining area immediately beneath the endothelium. We can't see an internal elastic lamina here because elastic fibers and elastic lamellae do not stain using hematoxylin and eosin. So we just have to guess where the boundary is between the tunica intima and the tunica media, and it's going to be somewhere just around here. From here to here is the tunica media of the vessel, and you can see smooth muscle nuclei, classic smooth muscle nuclei. If we looked at higher magnification, some of these would appear corkscrew shaped, which is a very classic appearance of smooth muscle nuclei, and these smooth muscle nuclei are circumferentially oriented, indicating that the cells are circumferentially oriented around the diameter of the around the circumference of the vessel. And finally out here we have a tunica adventitia, which is a layer of connective tissue, loose to moderately dense connective tissue. If you hunt around on the edge of the vessel, you'll see it also contains other smaller vessels like arterioles and venules, and it blends with the connective tissue of surrounding structures. So it's difficult to say exactly where the tunica adventitia of the vessel ends and where other connective tissue structures surrounding begin. But it should be pretty apparent to you that the thickest part of the wall of the artery is the tunica media, extending from here to here, composed of circumferentially oriented smooth muscle. Next we'll look at the equivalent vein, which is on the same slide. Here's the accompanying vein. Here's the lumen. The vein has collapsed down on itself, and this is because the thickness of the wall is small relative to the lumen size, and so it's not a very stable structure unless it's filled with blood. The endothelial layer which lines and is the inner surface of the tunica intima is along here, and we can see endothelial cell nuclei abutting on the lumen. The tunica intima, it's difficult to make out precisely where the tunica intima begins and ends, and arguably we could say that the tunica intima really only extends this short distance here. Then we have a tunica media, which extends variably from, say, here to here, or on this side from here to here, and the tunica media is proportionally made up mainly of connective tissue. However, it's important to note that there is smooth muscle present. These are bundles of smooth muscle cells, and where the smooth muscle is present, it's cut in cross or oblique section, indicating that these smooth muscle bundles are oriented longitudinally with respect to the long axis of the vessel. And this is a significant difference between the organization of smooth muscle in large veins versus that in large arteries. The muscle doesn't form a continuous longitudinal layer or indeed a continuous longitudinal bundle, but as we can see, it's scattered islands of smooth muscle oriented longitudinally, in between which are very large amounts of connective tissue. And although it's not apparent on this slide, the tunica adventitia begins here, and in most veins, other than the larger veins such as this one here, the tunica adventitia is in fact the most prominent layer of the vein. Large veins too have vasa vasorum, and we can see some of these here, and in fact if we looked here I think we'd probably find some peripheral nerve as well. Here we're looking at that muscular artery having been stained to demonstrate elastic fibers and elastic lamellae. The first thing which is apparent is the very, very dark staining of the endothelium and tunica intima, and in fact this is partially due to the endothelium and intima itself, but principally due to the thick internal elastic lamina which is present here, and indicating that there's a very, very thin tunica intima just inside or internal to that. So the tunica media extends from here to here. Along here is the boundary where the external elastic lamina would be located, and so what we're looking at in here are circumferentially oriented smooth muscle cells, the collagen which is deposited between these cells which doesn't stain and we can't see here, and the elastic fibers which do stain with this elastic stain and which are found interspersed among the smooth muscle cells. Interestingly, when we get to the tunica adventitia, as you can see here, the number of elastic fibers, albeit that they're small elastic fibers, increases, and so there's a more elastic adventitia here than there is media, and this is another characteristic of muscular arteries when they're stained to demonstrate elastic fibers. Here's the accompanying vein stained with the same elastic stain. Again, we have the endothelium lining the luminal surface here. The first thing you'll notice is that there isn't any dense, dark, intense staining internal elastic lamina, and neither do the tunica intima or endothelium stain intensely. And the next thing that you'll notice is that the material which does stain in the tunica media doesn't appear as long fibers really, but rather as small punctate dots. And this is probably indicative of elastic fibers which are oriented with their long axis paralleling the long axis of the vessel, so they've been cut in cross-section in the tunica media. However, as we move out to the tunica adventitia, as we would see out here, we start to see short, occasionally branched, thin elastic fibers present in the tunica adventitia, but not as many as you would find in the accompanying vein. So you can look and take some pictures and make comparisons for yourself between the muscular vein and the accompanying muscular artery with respect to both the arrangement as seen in H&E and also the presence and arrangement of elastic fibers as seen with special elastic stain. Here we're going to take a look at the largest vein in the body and the wall structure of the largest vein in the body, and this is the inferior vena cava. And what we're going to look at is the tunica intima. We can just make out maybe where the intima is here, the tunica media, and then the adventitia. And as before, at low magnification, there's very little detail we can make out. So we'll move to higher magnification to see how the structure of the inferior vena cava differs from that of the aorta. This vena cava has also been stained with elastic stain to demonstrate elastic fibers, so it's a good comparison to make with the aorta slide which we looked at previously. Here's the luminal surface, so it's lined by endothelium, which is not terribly distinct, although it's present on this particular specimen. The tunica intima extends from the tip of the arrow here to here, and in fact there is an internal elastic lamina. We can just make it out here. It's 2 to 3 layers of elastic lamellae. One thing you may notice is the connective tissue that forms the tunica intima. It's quite thick, but not quite as thick as you would find in the aorta. And there are these cells with elongated, sometimes spiral looking nuclei, although it's not evident at this magnification. Most of these are fibroblast-like cells, but there are occasional scattered smooth muscle cells found in the tunica intima. Here's the internal elastic lamina, as I mentioned, and one key feature to notice here is immediately inside the internal elastic lamina is a layer of smooth muscle. This happens to be, and as we look at it we can see the smooth muscle cells are cut in cross-section, and this indicates that they're oriented longitudinally with respect to the long axis of the vessel. And this is a feature of the inferior vena cava, is this internal layer of smooth muscle oriented longitudinally with respect to the long axis of the vessel. At higher magnification we see the surface on which the endothelial cells would be found. There's an endothelial cell nucleus here and here. Beneath that we can see the connective tissue, fairly loose, uncompacted connective tissue initially of the tunica intima, followed by denser, more compacted connective tissue with these elongated fibroblast nuclei compressed between plates of collagenous material, followed by the internal elastic lamina at the tip of the pointer here, and then, as I mentioned, this internal layer of longitudinally oriented smooth muscle, which means that here it's going to be cut in cross-section. As we move to the tunica media, which is going to extend from here out perhaps to about here, or maybe perhaps a little further. What we see is the tunica media looks largely collagenous, dense irregular connective tissue in nature, and interspersed among this collagenous connective tissue are fibroblast nuclei, which we can see here, the elongated ones. There are also some nuclei which may belong to scattered or dispersed smooth muscle cells, this one here for example, and in fact there are scattered and dispersed smooth muscle cells found in the tunica media, but they're not terribly well organized and there aren't too many of them. And there are a small number of elastic fibers, generally thin, sometimes branched, that are found in the tunica media, and you can compare this with the disposition and size of the elastic fibers or elastic lamellae that are found between the smooth muscle cells in the aorta in the tunica media. I've reduced the magnification here again. When this specimen was prepared, the tunica adventitia was largely separated off from the tunica media and the tunica intima, so there's not much adventitia present. The boundary between the tunica media and the tunica adventitia occurs along this line just about here, and once we enter into the tunica adventitia we can see vasa vasorum. This is a small arteriole here, and there's a venule here, and I'll leave it up to yourselves to look around the rest of the length of this particular section of inferior vena cava to see what other features you might be able to see or note.